Our topic today is a gel electrophoretic method, which allows us to assign protein spots coordinates of proteins separated in 2 dimensions using a gel electrophoretic procedure with much better confidence and reproducibility than was possible before. I always like to start by introducing the people involved. The research was based on a collaborative project with a small company from Cologne called Luftqualität und Raumklima, which is interested in the detection of airborne allergens both at the workplace and in households. Dr. Grün, the CEO of the company, convinced me that it is very helpful in occupational medicine to visualize dust proteins using 2-dimensional gel electrophoresis, with the idea in mind to detect protein workplace allergens early. We have had 2 graduate students working on the project. One was Wei-Chin, who just finished, and Marina took over. Benjamin was a diploma student, and Doreen is a very skilled technician, highly involved in the project. On the side of the company, there was also Birgit doing some data analysis, and Markus Müller is actually not a member of the company, but a self-employed informatician helping with programming. The project was funded by the Ministry of Economics and Technology in Germany. So samples would be collected from bakeries or mills or other workplaces with allergenic potential and checked for known allergens such as globulins, alpha amylase, etc. The point here is that there are still no defined limits of exposure for airborne protein allergens yet. So if a worker starts to suffer from work-related asthma, hay fever, or neurodermitis, he or she needs to be removed to an allergen-free workplace. So the task for our lab was to first isolate and separate the dust proteins in 2 dimensions and detect their coordinates as reproducibly as possible, with the ultimate goal of adding them to a database which should be available online for referencing. But not even the first part of the task was simple, because dust proteins come along in a package with soot and grease, and there's not much protein in dust anyway. So they were just horrible to purify, but that is quite another story. For the assignment of the spot coordinates, we initially suffered from the delusion that we could sort of work with the established Deutsch technology. This technology has been around for some time, and we have the entire pipeline available in our lab. But our experienced technician, Mrs. Berg, made it soon clear that there were a number of problems which the Decider software, which is programmed to analyze the gels of a Deutsch project. The software was not able to handle our gels. In particular, the fact that we received singular samples, biological replicates, which are the very basis of Deutsch, were in our case not available. But what we did want to do was work with the concept of using relative fluorescent dyes for parallel labeling of sample and standard. In order to be able to do that, we had to come up with a reference gel / proteins to which the sample proteins could be linked for coordinate assignment. So our collaboration partner was providing a bioinformatician who was supposed to program the algorithms to do that. What we needed, however, were reference protein spots distributed evenly across the gel. But everything we created was not suitable. We started with commercial reference mixtures, such as this one shown here, which provided too few spots. Then we tried to mix our own mixtures. And we even cross-linked proteins in an effort to improve spot density. We also thought of using whole proteomes, just E. coli or yeast or something like this, for referencing against intense spots. But even that had its downside. So, and just to mention the efforts behind that, with signals like represent smallest months and months of trying things and not getting anywhere. Eventually, more or less out of desperation, we decided to ignore common wisdom and lay a grid over the gel. It certainly would make our programmer happy. He could use triangulation to hook up the sample proteins to the reference. We figured that we could use features of 1D page. For one, we needed spots instead of bands, so that we trimmed commercial combs to provide V-shaped teeth. So you can see the bio-red combs here. We took scissors and cut them to a V-shape essentially. As you can also see, there are different ways of doing that. They all have advantages and disadvantages. If the angle is too, it depends on the angle. Sometimes the wells might collapse. You also don't want too wide angles when the spots will not be formed very well, and so on. We had to show that this works. You can see here we used a comb with both rectangular teeth and V-shaped teeth, and it is obvious that we get a nice spot shape here in this case, and the bands as we know them in the other case. So in order to use this for two-dimensional electrophoresis, we had to assemble the gel in a little different way. This is what it looked like. We have here the regular gel matrix. We are looking from the side now. On top of this sits the commercial PI strip. And then this is the trick. On top of this strip we pour another stacking gel where we insert the comb. We called this assembly gel strip sandwich. We proceeded to design our own combs, which served our needs much better and fitted much better in the tanks. We had them made in our workshop. The distance of teeth allowed the use of multi-pipettes, so this is then easier for sample filling of the wells. And the combs, which is also important, were adjustable in height, as you can see here, because you don't want to damage the PI strip while inserting the comb. We started out with polycarbonate as a material because commercial combs are usually made out of polycarbonate. We also tried Teflon. Again, there are advantages and disadvantages, but so far we like the polycarbonate best. As for filling in the reference proteins into the wells, there's also this little trick of increasing the glycol concentration so that the sample really stays at the bottom of the well and not diffuses to the edges. That provides much better spot shapes. So assembling the gels and the gels inside the tank required some skill. With Wang put the buffer chamber at an angle to do this. What we also needed were short polymerization times for the stacking gel so that we would not lose resolution in the proteome of interest. We are down to 15 minutes now by essentially doubling the amount of TAMET and APS. You will now immediately confront me with the argument of artificial protein modifications by residual monomer, which is correct, of course. We do not recommend our method for the purpose of protein identification. If you still need to identify the proteins in on your gel, you have to do it on a separate gel, although I'm not too pessimistic about getting an identification from our gels as well, but just for accuracy's sake, we don't recommend it. Next, we needed a protein reference mixture. which we generated from commercial proteins containing things like myoglobin, albumin, trypsinogen, and so forth, ubiquitin. It should provide spots across the whole molecular weight range, and the intensities of the staining should be about the same. What we also learned, what one should not put into the mixture proteins which would self-fluoresce, such as cytochrome C. We had named our method Comparative Fluorescence Electrophoresis, CoFGE, and now we had to show that it was any good. To that end, we had to do replicate experiments. So 3 gels each were run a week apart using E. coli as sample proteome. And you can see this here in red, and the grid was formed by our standard proteomes in green. As you can see, even when done by an experienced technician in parallel, gels could come out quite differently. Looking at those grids with waves here and joined lanes, one does wonder if the whole endeavor was pointless. For illustration, here is shown gel 5. This would be the proteome, well separated, and this is a grid run on the same gel. We typically labeled with Psi3 and Psi5 at this point. This slide just shows the whole scanned images so that you get an idea where the strip touches, what the wells look like, and for instance in this case we are missing one lane completely because obviously the well broke down. So this is some information which needs to be transmitted to the person who does the analysis later so that it doesn't start with the wrong lane for the grid. To generate a master gel, we averaged the coordinates of 12 grid gels and more or less hand drew the master using a ruler and then Photoshop or PowerPoint, whatever you want to use. In our laboratory we have PDQuest and we have also Decider, and we had difficulties with the analysis of this type of data. Our collaboration partner was responsible for the analysis part anyway within this project, and they had obtained delta 2D. So we sent the images to Cologne and we proceeded to warp them and match them to our sample grids. This is what it sort of looks like afterwards, the original gel and then the warped gel. We picked a number of landmark spots, 50 in fact, and determined their coordinates before and after this procedure to find out if the correction improved anything. There is a table in the paper we published in the meantime, so you can download it if you want to see all the data. Important for this talk is the fact that with no warping we have an average deviation here up to 7% in the Y dimension, and it goes down both in X and Y dimension to below 1%. And that was quite amazing to us and also to a number of other people. But we are still aware that we are ignoring the variation due to PI. And within this procedure we simply hope for exceptionally manufactured PI strips. They do seem to be quite good though according to those results. This was the state of affairs when we applied for the patent and wrote the paper published in 2012, so a number of the details, or a lot of the details you find in there. In the meantime, we have transferred the method to horizontal electrophoresis because we have this instrument from the company server, which is called the tower and which allows the simultaneous running of 4 gels. I'm showing you a layman video of our graduate student Marina working. I have to apologize for the quality of this video, but it certainly shows you what she needs to do in order to produce gel useful for this purpose. So we purchase the commercial gels from Server, and they contain a groove for the PI strip and a well for the marker. And what she has to cut out now are the spots for the reference grid. She uses a comb to locate the position, and what she currently is doing is to remove the gel plugs from the positions. So this will take a while, and in order not to bore you, I guess I fast forward here. So she is pretty much done now, and we get some sort of wells, round wells, which hold about 0.1 microliter of liquid, so much less than we use in vertical electrophoresis. And even though she has created the wells by hand, the gels which come out of this look quite striking, and Server was very pleased to see that. So again, we have E. coli proteome in red and the grid in green. And there's certainly hope Server is interested in all of this once they start to produce the commercial gels with wells made by some industrial procedure that everything will look much better again. So again, we had to run through the whole song and dance of providing usefulness. In this case, 4 gels each were run, and I asked her to provide me, by now, by the way, we are doing the analysis in Münster ourselves with delta 2D. So she has given me a few slides on how to do the analysis to match it to the master gel, the artificial ideal grid. Those are the experimental grid gels, and those are the proteome gels. This is what the correction of the match vectors looks like from the ideal grid to the experimental grids. And this is a particularly bad example actually of gel having a bubble or some unregularity here in the middle, and once it is corrected, it does show an improvement here. Same deal, in this case we have chosen 30 spots to do the comparison. If we use all 8 gels, again run 2 weeks apart, the experiments, then she improves the average of mean to about 1%. If she only uses 3 of the best gels, she is well below 0.7% in the improvement. So summarizing, we have applied our method both to vertical and horizontal gel electrophoresis. We are much in favor of the horizontal electrophoresis. It's much easier to do, but if you don't have it, vertical is fine as well. Ms. Marina has also tested the relatively new G dyes now with the method, and it works as well. Using those, she was able to show even lower detection limits than the manufacturer itself, using spots instead of bands. What we are currently testing is the use of a 3rd dye and 2 or 3 proteins across the PI range to hook also the PI up to the ideal grid and have this correction also. I can't show any data yet, but so far it looks good that we will have them in the soon future. So as I mentioned, all of this was published, and with that I'm finishing up my talk, and I invite you all to Münster for November for our annual, and this year the 10th conference on nanoanalytics and single cell analysis. I thank you for your attention.